Good evening, everyone. I welcome you to the IHERA Journal Club today evening, which is going to be on iron biomarkers in male infertility are associated with sperm DNA hydroxymethylation and cumulative live birth rates, the prospective studies. So here we are going to discuss this journal article, which is published in the Journal of Translation Medicine, which is an open access journal which accepts article related to human experimentation so as to optimize the communication between the basic and the clinical science. It is a high impact factor journal, which is having an impact factor of 7.5 and 5-year impact factor of 7, which is very quite high, and it is a open access and well-known journal. So as we know, oxidative stress and iron homeostasis are deeply intertwined with sperm function. Iron is a double-edged sword essential for normal physiology, but in excess, it contributes to ROS, sperm DNA fragmentation, and thereby affecting the fertilization potential. So we are very lucky and fortunate to have with us today the authors of this journal club. Having them here gives us a unique opportunity not just to review the findings, but also to understand their thought process, challenges, and future direction directly from the researchers themselves. So I welcome you all to the Journal Club. I would like to introduce the founder of IHERA, Ved Prakash sir, and Sanjay Shukla sir, the co-founder, and Charudat Joshi sir, who is also a co-founder. We are the co-conveners, Dr. Nidhi Singh, Yoshita, Nancy Sharma, and myself, Dr. Anantalakshmi. So I will be leading this JC along with Bala Kumaran. Sorry, due to an emergency, he is not able to join with us. Next, moving on to the to welcome the audience as such and the speakers and the authors of the journal club, I would like to request Ved Prakash sir to take over the session.
Thank you, Ananta. And first of all, good evening, everyone, and good afternoon, good morning, everyone. So this is very, that I started with the I Journal Club. Like we try to include all the authors, original authors who that published the article. So it will be great to hear that people can learn and experience the things what they have done in the publish the article during publication of the article, and what finding which can help us, and maybe we can also find new things we can use in our clinical practice also. So this time we are very thankful to Dr. George who accepted our invitation with Dr. Yomna and Dr. Chadi Faki. So I welcome all of you. And today the topic as discussed with as told by Ananta, that iron biomarker in men with infertility, which are associated with sperm DNA or not. So how that pregnancy will affect by these, or it will affect or not. So let us see that the finding what they will tell us about this article. So thank you all and welcome all of you. Thank you.
Thank you so much, sir.
Thank
Now,
you.
I'd like to introduce the moderators of this session. Professor Sanjay Shukla sir. Sir is a senior consultant clinical embryologist working as an adjunct professor in clinical embryology with more than 25 years of experience in the field of ART. I welcome you, sir. Next, I would like to welcome another moderator, Dr. Rajvi ma'am. Rajvi ma'am also has a vast experience in this field. She has an experience of more than 30 years, and she is an invited speaker at many conferences. And at present, she is a treasurer of Academy of Clinical Embryologists and chief editor of ACE newsletter. Ma'am, I welcome you, ma'am. I would like to hand over the session to you to introduce the speakers and authors of this session.
So, thank you, Anant Lakshmi, and congrats to IHERA for taking up this a very interesting topic, which actually helps us to understand basic science and its clinical applications. And it's something which we are absolutely, with the advent of ICSI, we are trying to forget the basics. And I really compliment IHERA for selecting, and definitely kudos to the authors who have published this. So looking forward to the discussion. So primarily, I would like to introduce Dr. Yamuna Murad. She's a medical doctor and a clinical pathologist who's graduated from AUBMC with 12 years of experience in laboratory medicine, a lab director at Al Hadi Laboratory and Medical Center, She has worked across multiple disciplines, which is something which is really commendable because that gives you the whole scope of lateral thinking from biochemistry, hematology, micromolecular biology, and genetics. And she's also directed the PGT lab in the different IVF centers. And we're looking forward to hearing from you, Dr. Yamuna, about the work, about this specific paper. So the next author is Professor Chadi Faki, and he's an associate professor at the Lebanese University and the director of Al Hadi IVF and Genetics Center, who has many publications. We are familiar with your work, sir, and we have also heard you at different platforms, and looking forward to your guidance, which has led to the paper. And yes, the third author of the paper is Dr. Georges Rad, a PhD in clinical and therapeutic research from Côte d'Azur, University, France, a scientific director at Al Hadi Laboratory and Medical Center, Beirut, Lebanon, and a lecturer at two of the top Lebanese universities. So we see a good integration of clinical application, the scientific aspects, and the academic aspects. So thank you so much. And over to you, Anant Lakshmi, to lead the
You can start the presentation. Thank you.
Yeah.
George, you can share your screen now. Yeah.
Can you see my screen?
Yes, just a minute. It's
Okay.
loading. Yeah, it is visible. Thank you.
Good afternoon, and thank you so much for this kind invitation. We are delighted to be with you today to discuss the findings of our newly published article entitled Iron Biomarkers in Man with Infertility are associated with sperm DNA hydroxymethylation and cumulative live birth rates, a prospective study. Let's start with a brief introduction about the topic. Iron plays essential role in the body for oxygen transport, oxygen storage, energy production, and also in spermatogenesis, because spermatogenesis is an iron-dependent process. After its absorption from the intestine, iron binds to plasma transferrin and walks through the bloodstream. Once inside the cell, iron is stored in ferritin. Interestingly, inside the testicle, Sertoli cell can synthesize transferrin, called testicular transferrin, to ensure a tightly regulated metabolism for spermatogenesis. Clinically, iron status can be measured by several markers, such as ferritin, free iron, and transferrin. In addition, we can measure the binding capacity of transferrin, called the unsaturated iron binding capacity, UIBC, and total iron binding capacity, TIBC. Let me tell you that UIBC is the measurement of vacant iron sites on the transferrin. Iron homeostasis is very important for the body function because iron deficiency or iron excess can lead to oxidative stress. And this kind of stress is known to alter sperm physiology at several levels. In addition, iron can be a cofactor of several enzymes, such as TET. TET enzyme stands for 10-11 translocation enzyme. It's an iron-dependent enzyme. It works by converting the 5-methylcytosine in the DNA into 5-hydroxymethylcytosine in the DNA. Methylcytosine and hydroxymethylcytosine are part of the epigenome, controlling gene expression. Why the epigenome is very important in reproductive medicine, and specifically for male infertility, because previous studies showed that paternal exposure to certain environments, such as high fat diet, smoking, alcohol, may alter the epigenome of the sperm. When we talk about sperm epigenome, we're talking about the chemical modification of the histones, the chemical modification of the DNA, and the activity of non-coding RNAs. Alterations in the epigenome of sperm after paternal exposure was associated with several diseases in offspring. Back to our TET enzyme. TET enzymes are successively expressed during human spermatogenesis, and their expression level is pivotal for male infertility. However, the influence of iron biomarkers on sperm DNA methylation and hydroxymethylation has not yet been fully investigated. Therefore, this study aimed to assess whether iron biomarkers in men with infertility were associated with global DNA hydroxymethylation in spermatozoa and with cumulative live birth rates. How did we proceed? We enrolled infertile couples attending Al Hadi Laboratory and Medical Center Beirut for ICSI. In-person interviews were conducted with these couples, and we've excluded from the study couples undergoing ICSI with embryo biopsy, frozen gametes, or with testicular biopsy. Also, we've excluded from the study couples where the female partner is obese, the age is more than 36 years old, or having less than 3 cumulus oocyte complexes on the day of oocyte pickup. At the end, we included 60 couples where we fixed the female factors. All the female partners underwent a controlled ovarian stimulation, and on the day of oocyte pickup, the male partner provided a semen sample and a blood sample. Each blood sample was divided into 3 aliquots. The seminal fluid free from cells was used to assess iron biomarkers. Also, the serum from the blood free from cells was used to assess iron biomarkers. The second fraction of the semen was subjected to density gradient, and the motile enriched sperm fraction was used for ICSI. Embryos were cultured for 5 days in the lab, and cumulative live birth rate was calculated. The third fraction of the semen was also subjected for density gradient, and the motile enriched sperm fraction was subjected for DNA extraction, and the extracted DNA was used for sperm global DNA hydroxymethylation assessment using ELISA technique. Let's see the results. As a recap, the primary aim of the study was to assess the association between various iron biomarkers and the global DNA hydroxymethylation levels in spermatozoa. How we assessed iron levels? So first of all, iron Fe3 plus were separated from transferrin using citric acid and low pH. The free Fe3 plus were reduced to Fe2 plus using ascorbate. Ascorbate formed a colored complex with ferrosin, and the density or intensity of the Interestingly, we found a significant positive correlation between serum iron and sperm DNA hydroxymethylation. In addition, we found a significant positive correlation between seminal fluid iron and sperm DNA hydroxymethylation. Then we measured the UIBC, which is the unsaturated iron binding capacity. So, as I told you before, UIBC measures the vacant iron sites on the transferrin. So, to measure this vacant site, we've added a known concentration of iron to transferrin. After saturation, the leftover or the remaining iron were reduced using ascorbate to Fe2 plus. Fe2 plus formed a colored complex with ferrosin, and the intensity of the color reflected the concentration of the iron remaining. Interestingly, we found a significant positive correlation between seminal fluid UIBC and 5-hydroxymethylcytosine percentage in the sperm. Then we measured or we calculated TIBC. TIBC, it's the sum of iron plus UIBC. TIBC in serum and in seminal fluid were positively correlated with 5-hydroxymethylcytosine. So, to sum up this first step, we found that serum iron and TIBC on one hand, and seminal fluid iron, UIBC, and TIBC are affecting or related to 5-hydroxymethylcytosine in sperm DNA. Then we've included these variables in a multivariate analysis to quantify and to check which one is more important in the influence on 5-hydroxymethylcytosine. The multivariate analysis showed that serum TIBC demonstrated a statistically significant positive association with sperm DNA hydroxymethylcytosine level with a p-value of 0.02. Now for the secondary aim of the study. The secondary aim of the study was to determine if iron biomarkers in serum and seminal fluid were associated with cumulative live birth rate in infertile patients undergoing ICSI. To address this, we used 3 steps. First step, we compared iron biomarkers in serum and in the seminal fluid. Among the markers that we measured the transferrin levels using immunobeads. The immunobeads would form precipitate, and we can quantify the amount of transferrin in the fluid. Our results showed that systemic iron regulation is partially reflected in seminal plasma. Why? Because the seminal fluid transferrin was positively correlated with serum transferrin, serum TIBC, and serum UIBC, with a p-value of 0.004, 0.007, and 0.008. Next, we wanted to assess if iron biomarkers in semen and serum are associated with sperm parameters. We found that serum iron, transferrin, and TIBC were positively correlated with sperm motility. In parallel, seminal transferrin was positively associated with sperm count. Third, we assessed the impact of iron biomarkers on cumulative live birth rate. How we calculated in this study cumulative live birth rate? It's the occurrence of at least one live birth from a single ovarian stimulation divided by the total number of embryo transfers, fresh and or frozen, derived from that cycle. We found that we have 1% increase in cumulative live birth rate per 1 milligram per ml increase in seminal fluid iron. However, we have a 3.7% decrease in cumulative live birth rate per 1 milligram per ml increase in seminal fluid transferrin. Let's discuss and try to understand these findings. From iron flux to DNA hydroxymethylation and mechanistic insight. So when the iron level in the body is not always available, or when we have a deficiency in iron, the body tries to adapt by increasing the circulating transport capacity. So the body tries to increase the amount of transferrin, TIBC, and UIBC. What is the purpose of this increase? To ensure a proper or sufficient amount of iron for the intercellular process. In this case, we found that an increase in iron can increase the level of hydroxymethylcytosine. However, increasing hydroxymethylcytosine, is it good or bad thing? We don't know, and further studies are needed to understand what is the impact of this increase in 5-hydroxymethylcytosine on the health of the offspring. Yes, there is a compensation, but it may come at the expense of sperm quality and outcomes. Due to iron fluctuations, at a certain point, sperm can suffer from this adaptation, and DNA fragmentation maybe can develop, or other alterations may happen in the sperm that we didn't measure in the study. And these alterations were associated with decrease in cumulative live birth when high transferrin was found on seminal fluid. As conclusion, these findings suggest that monitoring and managing iron levels may play a crucial role in male fertility. To establish a causal link, RCTs are needed to investigate the impact of iron on semen quality, potentially contributing to the development of dietary guidelines for men seeking to optimize fertility. At the end, further studies with a larger cohort of infertile patients are recommended to assess the long-term effects of iron status and sperm DNA hydroxymethylation exchanges on offspring health. Thank you for your attention.
Thank you, Georges, for a very clear, crisp presentation from the basics. Really appreciate it. Maybe I'd like to open the floor first before I start off. If there are others who would like to, Anant Lakshmi, you would like to start the discussion? You're on mute.
Sorry, ma'am. I just want to know why they have selected the motile sperm fraction alone for the study instead of taking the sample as such. Yes.
Dr. Shadi, okay. So since the semen contains various sperm population, motile, non-motile sperms, and dead sperm, so we wanted to check the impact of iron on motile sperm, which are involved in fertilization and conception, because non-motile and dead sperms has no chance to reach the oocyte. We want to mimic a bit the nature, not to have an effect diluted in the whole semen sample.
So you have not used any preparation method, just select swim up alone you have
No,
chosen?
they did density gradient.
Okay.
We did density gradient.
Okay. So will that preparation won't influence your outcomes as you're studying the sperm DNA fragmentation, hydroxymethylation and all? Won't you think the preparation method will increase the fragmentation as such?
Yes, we published a paper before showing that sperm preparation techniques may alter the quality of the sperm. Dr. Shadi, can you help me?
Yes.
So we showed that before in a publication before in embryology that sperm preparation techniques may influence. It will be interesting to check if the iron status in man, we have to select a particular sperm technique for them.
Thank you.
So, I would like to go back to the thing. The first thing is, when we do know that iron plays a major role in spermatogenesis, what about the male partners? I mean, were these all normal normospermic partners? Because your data doesn't show, and the mean is nearly 50 million. So naturally, you took only with normal normospermia, or
Yes,
you
we took
excluded
patients
any with severe oligospermia?
We excluded all patients having less than 5 million spermatozoa per ml.
Okay, so there were
So 5 million and above.
Okay, so that means you had a mix. So was there any kind of relationship, because since it affects spermatogenesis, if you obviously would have the data on association between sperm concentration versus the different iron binding levels and total iron.
Yeah.
Would you that, because that would sort of explain even the causes of oligospermia, whether it is deficiency in iron or iron transport or iron uptake, which is responsible for it. So you have some data on that, because that will be very interesting to see that one is systemic iron deficiency. Second is whether that systemic iron deficiency is being reflected in seminal plasma iron deficiency, or is it associated with spermatogenesis and leads to oligospermia, since you did have patients with oligospermia, although you didn't take anybody with severe oligospermia less than 5 million. So that would also be an interesting finding, and whether iron supplementation subsequently would help in these patients. So maybe your views on that, or you have some ideas or what could be.
Dr. Shadi.
Yes, iron deficiency in man is variable from country to country. In Lebanon, we have around 5 to 10% of our male population in Lebanon has iron deficiency. So it is a high number. In USA, it is around 0.2%. I don't know in India if you have any idea about the prevalence of iron deficiency in man. In Lebanon, it's not lower. It's not so lower. So and the correction of iron deficiency, as it has been shown in this study, has an impact not only on assisted reproductive techniques results. It may have an impact also on future generations.
Right.
So And I think that this study, the main good point in our study, it's an eye opener for other studies, mainly in countries where our men have iron deficiencies.
No, I agree with that. I think that's very interesting, and it is useful because then the treatment is much more simpler. And if you can really see the association between iron deficiency, and if that improves spermatogenesis or even the quality, then I think we have a big leap. So maybe if you can look at your own data, it'll be interesting to see whether it is, there are low or high transferrin levels which would sort of reflect iron deficiency, and then see whether you're having a low sperm concentrations. That would be. Also, what is surprising is you had a very high percentage of population who are smokers, nearly 75%.
Yeah,
And
Dr. Rajvi, I was also
stunned with
wondering,
that.
and
Yeah.
in fact, I have just marked all those things here
Oh, you're back.
because
Yeah.
it is well
It's
known
very
fact.
high.
I mean,
Yeah.
there are several studies which show the correlation between iron deficiency anemia and smoking. So I was wondering, even equal number of women, they are smokers, not just the men.
Yeah.
So
And it's
Dr.
pretty
Shadi
high.
tries to fight
75% is
for,
pretty high.
yeah. Dr. Shadi tries to fight against smoking, but in Lebanon, the culture, they use shisha, they use a lot of things to smoke.
Yeah, 50% of Lebanese women take shisha. 50%.
Did you
It's a
also
sig-
look at anything with the DNA fragmentation, since they were already exposed to such high levels of potential oxidative stress with such high smoking and all these? So,
But
or you don't value the DNA fragmentation test?
first of all, Dr. Shadi tries to push them to force them to stop smoking. If not, you stop them since 2 months, doctor. You ask them to stop smoking, but they
Must
don't
be
stop.
tough, because to get them to
Oh.
stop smoking, I don't know how easy it is.
Yes,
It's
all
not-
our
you can
ART
tell us,
population stop smoking at least 2 months before ART. I hear ART in Lebanon is paid by not by the government. It's paid by the people. So, and we tell them if you don't stop smoking, the results will be at least 1/2. So the majority of them, because they have to pay, they stop smoking at least 2 months before assisted reproductive techniques. But we still have the problem of iron deficiency.
Yes, Sanjay.
There's one thing, Dr. Rad, I would like to bring your attention to is there is a disparity in the number of women. It is mentioned that the all participants number is 60, but here it is like if you go to this smoking status, there is discrepancy. There are 27 non-smoker and 28 smokers. So it comes to 55 only. So there is no data
Five
about 5.
non-reported.
Yeah.
Yeah.
And also in the main section also, non-smokers are 21, which is correct, like 35%. But if we see 39 men, so it should be 65%, not 76%.
Okay. Yeah.
So it needs to be corrected, I think.
Okay. Yeah.
Also, the paper, I mean, it's very nice that you're actually going to clinical live birth rate, which very rarely people do manage to follow up. And because you're concerned, worried about the developmental origin of disease. Did you also look at the embryo arrest data, number of embryos which arrested on day 3? Because that would also be a partial reflection of the sperm quality and especially sperm function.
Yeah. So we have this data, but we didn't calculate. But you opened my eyes. Now we will look and calculate to check.
Yeah, because that, the couple of things what I find in the paper, which you would definitely have data, is to look at the, the iron levels, seminal plasma, and the sperm concentrations, because you do have oligospermic. So, and you have a good mean of 50. median of 50, I
Yeah.
think. So you have a good median. So you'll
Yes.
have many who are having a good sperm concentration, and see
Yeah.
the association between sperm concentrations. That would be quite interesting. And that would be, and especially with also the transferrin. So if transferrin is high,
Yeah.
binding
High,
is
yeah.
low, you sort of get a clear idea whether it is the circulating iron deficiency, especially since it's 5 to 10% in your country. And I think in a lot of countries, even India, may be high iron deficiency. I don't have the data on hand, but that would be a very good idea to say whether just simple supplementation would help. Is it the dietary changes, absorption changes, which is leading to, we are all seeing a very rise in male infertility all over the world. So, it will give us, open our eyes to have a wider look. And also the arrest part, I think you can have, because obviously you'll have data on the embryo arrest and development, how many went on to blastocyst or conversion rate. So that will also tell us whether, the quality of sperms. It's not just quality, because you're looking far beyond. We're just otherwise looking at sperm count, motility. You're actually looking at iron, and you have given, we know the association, and you presented it very well, the role of iron in spermatogenesis and subsequent. So that will be nice if you could look at that too.
Yeah. We've reported the relation with fertilization and vasculation. However, we didn't report with the embryo arrest, but we have the data. We can look
You
and
can
check
look
what,
at it.
what
It will
happened.
be interesting.
George, let me add something, please, concerning, I want to highlight on the transferrin in seminal plasma. As you know, transferrin is produced in a very high percentage by Sertoli cells in the seminal fluid. So most of the transferrin in the seminal fluid is produced by Sertoli cells and not serum derived. So it's important for later studies maybe to study the testicular transferrin versus the serum-derived transferrin and its impact on sperm parameters and hydroxymethylation. So this is an important point.
Can we detect particularly testicular transferrin? Is there a technique
Yes, it's
to
a
detect
different
particular?
isoform. The technique is a little bit more complicated because we have to do a blotting technique to separate the different isoforms, but it is feasible.
But
So we
the
can separate and
paper,
quantify.
because the testicular or seminal plasma transferrin was very low compared to what you have in the serum. Serum was nearly 280 versus seminal plasma was 1. So is it that the different isoforms did not get measured, or it doesn't matter? I think you are the ones who's an expert on transferrin measurements.
The transferrin levels in seminal fluid, it is within the measuring range of the assay. That's why we could measure transferrin accurately using our Roche method, because the transferrin, it is the measuring range, it is between 10 and 520 milligram per deciliter. So we can easily measure the testicular transferrin, even if it is as a percentage, it's lower than the serum transferrin, but it's measurable. And the main transferrin isoform in seminal fluid, it is produced by Sertoli cells. So the measured transferrin, it's the whole transferrin level in seminal fluid, not only specifically the testicular one.
And what would be the amount, do you think, the transferrin from serum enters into the seminal fluid? Does it really, is there diffusion, or there is no? Will the transferrin from serum actually enter, or you don't think it's contributing to the seminal plasma transferrin at all?
The main transferrin contributing to seminal plasma is produced by Sertoli cells and not from serum.
Okay.
So the serum is around 30% of the total seminal transferrin. Yeah, the serum transferrin is 30%. 70% are produced by Sertoli cells. That's why, and there's a lot of publications talking about transferrin levels in seminal fluid and azoospermia. They found that it's very low. The transferrin level in seminal fluid is low in patients with severe oligospermia and azoospermia.
So that again highlights what we are talking about, looking at thing and maybe severe oligospermia and functional compromise. Also the ferritin levels.
Maybe the affinity of the transferrin, of the seminal fluid transferrin, the affinity to give the iron to the TET enzyme, maybe it's different. We cannot there should be more studies about how
Correct.
this transferrin differs from the serum by giving the iron
Binding.
ions to the So
Also, the
that's-
ferritin levels were very high. So what is the source for ferritin? Is it local or is it also systemic? Because
ferritin it's systemic.
it's systemic.
Yes, there is no local
production.
production.
But then the ferritin levels in your paper were much higher in the seminal plasma as compared to. So I would have presumed the reverse, that transferrin is systemic because the levels are much lower in seminal plasma, whilst ferritin is much higher in seminal plasma. So I would have presumed, with my limited knowledge of biochemistry to that level, that actually ferritin is more local. So it appears to be contrasting. Like, why would there be so much of accumulation of ferritin out there? It's any understanding what could be happening?
It's liver produced, the ferritin. So
It's exactly.
Iron.
Maybe because it's a male population, and usually the ferritin stores are high enough.
Yeah.
I have no explanation.
Usually it's higher
Yes,
than
of
women.
course.
No, because the same patient, you have serum
Wow.
levels
Same
in your
patient.
data itself.
Yes.
I mean, your
Yes.
data is in sync with the average found. So there's no major. But if you look at the serum levels and seminal plasma levels, it's so much higher. So I was just curious to know why it has happened, and why is it accumulating there?
Maybe because the sample was concentrated when we selected the semen, George, by doing the gradient technique.
Classification, yeah.
Maybe it's a
so maybe-
concentration. Yes.
So maybe you'll have to recalculate that also.
Yeah.
I don't know what are the ways, because
What's that? Yes.
yeah, Sanjay, over to you.
Did you also try to study or try to measure simple hemoglobin levels in these men?
Hemoglobin CBC levels, we didn't check for CBC in those men.
Yeah, because
For
if
anemia.
if we can find any correlation between the total hemoglobin levels and sperm DNA hydroxymethylation, probably this would be the easiest marker to do.
Most of males, they are not anemic. So the iron, also the iron level, if you can see in our population, we don't have a serious iron deficiency. As you can see, we are on the lower limit of detection. So the iron level in men, it's between 33 and 193 microgram per deciliter. So our iron, we don't have much below 33. The data is not with me now. George, do you have,
Yes.
we don't have a lot of iron levels deficient, less than the normal range. We don't have
They are on the
iron
lower
deficiency.
limit.
We don't have iron deficiency. So
No.
that's why we didn't check for anemia.
And if we look at the literature about iron deficiency, about anemia and abnormalities in spermatogenesis, we should have a hemoglobin level less than 6 to have alterations in spermatogenesis. So I think the hemoglobin level or the anemia should be very severe to affect spermatogenesis. It was not the objective of our study.
So when you
What is
mentioned
interesting-
5 to 10%, what was that? You mentioned that 5
to 10% of iron deficiency in
the population.
In the male population.
In Lebanese man, but not in our population, in this sample. The
Because
studies
we
in
don't
our
know.
laboratory showed that we have a ferritin deficiency in Lebanese man between 5 to 10%.
Okay,
Okay.
I
ferritin,
want
not
to add
iron
something.
binding.
I'm not talking about iron deficiency. I'm out in the serum. I'm talking about ferritin deficiency.
Let me add something, Dr. Shadi, please. Usually, before reaching anemia, first marker to be decreased is the ferritin, then the iron level, then the hemoglobin. So here we don't have anemia because we don't have a decreased
Okay.
iron, yani significant decrease in iron levels. So all the iron in our population, on
It's
our study
subclinical.
population, are within range, but we have a variation over this range.
Yeah.
Maybe that would
So,
also be interesting for you to look, because it is a range defined for other physiological functions. But if it is on the lower side and it's affecting spermatogenesis and other functions, maybe the stress and live birth rate. So maybe the levels required for spermatogenesis, presuming
Yes.
that it's also affected, and quality of blastocysts or implantable blastocysts, if it is something, again, it could be a very interesting to see that, because if that
Yes.
is there, so even if it may not be anemic, like Dr. Sanjay said, even if it is on the lower level somewhere,
Yes.
it may not
So
reflect
we
in
need two
anemia
cut-offs.
classical.
Yes.
Yeah.
Yes.
Yeah.
We need a cutoff on the low level.
Yes.
And even a high iron, it's also toxic.
Correct.
It will induce oxidative stress. So
Correct.
also we need a higher cutoff.
Yeah.
Yes,
I
it
mean,
was a low sample size study. We cannot draw
Yeah.
conclusions for
True.
phosphorylation rate, for cleavage rate from only this low sample size. So it was not the main objective of our study.
No, the study is appreciated. It is just stimulating a lot of thought.
Yeah,
What
Developing
else
ideas.
can be done? So, it's a
Developing
great
ideas.
start. I mean, these
Yes,
are just
yes.
ideas that come in, and you already have a lot of data and you all the expertise to assess. And maybe if you look at it, you're going to give solutions to us. So, I mean, this is like beyond the studies with the data you already have, and so it'll be very interesting
It's easy
actually
for
see.
us.
Yeah.
Yeah. It's easy for us to do it because we have an IVF lab and a medical lab.
Yes.
And
I mean, I can
scientific
see
lab.
that in the discussion. So it'll be very
Yeah.
nice to actually put in all this and give more solutions to the world,
Dr. Yumna, I'm curious to know what is the status of diabetes in your country?
What's the status
Country.
of what?
Diabetes in our
Diabetes.
country.
Diabetes.
Diabetes in
Diabetes
our
in our
country, in Lebanon.
It is
In
similar
Lebanon.
it is similar to the
6% in
international
Lebanon.
Canada.
percentages.
Mhm.
I will give you exact
6% in
numbers
Lebanon.
because we had a study done concerning this. We have a lot. Let me just show you something.
Type 2 diabetes in Lebanon, it's around 6%.
Because we have high
6%
prevalence of
overall,
obesity.
diabetes type 2 and type 1.
Mhm.
Let me give you some numbers.
Yeah, because once I heard about the Gulf countries and Arabic countries that
Yes.
they have slightly higher occurrence of
In the
diabetes.
Gulf, yes. In the Gulf,
Yeah.
they have higher percentage, yes.
And
They are
in Lebanon also we have high
obese.
prevalence of obesity.
Yeah. So because all these things, they are interrelated, they metabolic
Yes.
disorders
Yes.
and so I mean you have opened a whole lot of
ideas and and looking at male infertility in a totally different perspective. I think it's a great study, which
actually,
is a springboard for many things to do.
The very first question that came to my mind was, how did you think about studying this particular biomarker?
Yeah. So same thing I
What
was going
prompted
to
what
ask
prompted
them.
you to do?
Yeah, same thing I going to
We
ask
were
them why you started with this study, because iron biomarker we not able to think about this this will affect this kind of DNA fragmentation or these things. So nobody
if
is
you're
studying
follow,
actually, I think.
if you're
So
following a bit our studies before, we worked a lot on the exposure of man to environments and how it affects epigenome. While reading about this topic, we found the TET enzyme and its activity and how it works, and no one looked particularly if iron can regulate that activity. So
Long back,
there,
Dr. Rajvi has done a beautiful study on the effects of pesticides and insecticides. So
let me give you the numbers, the exact percentages. We have around 8% of diabetes in Lebanon, 0.1% of the population with type 1 diabetes. In Beirut, we have higher percentage. It's up to 12%.
Something
Type 2,
similar
Dr.
to
Ina?
India,
It's
actually.
type
Even
2?
India, yeah.
Yes, it's similar to international numbers.
It's type 2, Dr. Ina, or type 1, you said? Type 2?
كلهم overall
No,
8%.
overall.
Type 1,
8%.
it's 0.1%. Usually type 2, it's 90% of
Yeah.
all diabetes, and 10% are for type 1 percentages. بس I'm بحكي about the population, it's 0.1% type 1 and 8% overall.
Because they say India is around 8.9%. So that is also pretty similar. And in India also maybe urban population may be higher. The dietary lifestyles and stuff. So
Exactly. Yes.
What about
Do you have-
PCOS in your country? Do you have too much PCOS in your country?
Oh, yes.
Yes. In Lebanon, it's around 18%.
I don't think we have data. Do we have? But surely it must be equivalent. I mean, I think it's a worldwide phenomenon now. It's like you have kids in the 16 and 18 know about PCOS. It's not just the infertility specialist or gynecologists, or I mean, teenagers talk about PCOS.
Yeah.
So it's something which is alarming. It's
Dr. Shadi, why do you think the prevalence is increasing of PCOS?
It's a multifactorial thing.
George, what was your question?
Why the prevalence of PCOS is increasing worldwide?
It depends on the criteria to diagnose PCOS.
Criteria is very different also from country to country. We don't have the, not all the countries adopt Rotterdam criteria. Okay, you have the Androgen Access Society criteria, we have NIH criteria, we have, even in Europe, if you use, in Western Europe, if you use Rotterdam criteria, only 12% of the population has PCOS, while if you adopt the AES, the Androgen Access Society criteria, it will be around 30%. The prevalence of PCOS, yes, the discrepancy is mainly related not only to obesity, to the difference in obesity profile from country to country, but also to the criteria that you are adopting to define PCOS.
No, I agree with you, sir, on that there is the definition. But if you look at it from a very overview and that kind of a perspective, there's no doubt that there is a rise. I mean, it's not just that we are being extra critical, or, like I remember because I've been there in the field for quite a while, that initially you barely saw PCOS. It was more like textbook. Then they started coming in the late 90s. And one did think that it was, because of the transvaginal ultrasound and people are just assessing more. And then, of course, now we know there's no doubt about it, whether you classify it, maybe there may be different categories, insulin resistant ones, the lean ones, or the various factors. But irrespective of the criteria, overall, metabolic syndrome and associated PCOS is surely on the rise. And somewhere it seems to be running in parallel with the decrease in or the increase in male infertility. And there seems to be more of environmental factors, I think, which are various, and maybe all of them together, including lifestyle and everything. Because how do you just explain this exponential rise? Like something's there. I don't know. Maybe we need to, and it's difficult to study. I mean, how do you really study environmental? All you can do is associational studies or do some stuff in animals. But maybe ideas on this would be welcome, and collaboration on this is welcome to really look into it. And especially now that you have started looking at mechanisms, again, could be environmental, like you say, iron part.
We have one question from the audience. How do sperm DNA damage pattern differs between men with iron deficiency anemia and those with iron overload? Example, hemochromatosis. Did you have any specific pattern? Do you observe like that? They asked.
In iron deficiency and in excess iron, in both cases, we can have the status of oxidative stress, and OS can induce oxidative damage for sperm. So easier way to check the DNA fragmentation to check the oxidation of fragments and of the DNA. We didn't check in this population, in this study. Maybe later on we can do something to check. But there are several papers showing that iron deficiency and excess iron can lead to the DNA fragmentation due to oxidation.
Also, iron accumulates in patients with hemochromatosis. The main problem is the accumulation of iron in the testicular tissue, and this will impair testosterone production and sperm maturation. So it's not just an oxidative stress; it's a deposit of iron in the tissue. But also, oxidative stress with the high iron concentration is also it will impair deposit, mainly deposition. Iron deposition is mainly in this population, the hemochromatosis. We have iron deposition in testicular tissue and even in pituitary gland. Yes, so it will affect the hormones and the sperm maturation.
So is there any reference range so that can tell us about that that patient will be have more sperm DNA fragmentation or normal kind of things? Any reference range of iron biomarker?
For the DNA fragmentation?
Yeah.
George, for you.
So
we don't have a cutoff.
There has no cutoff because a few studies worked on iron deficiency and DNA fragmentation, and few studies. So I'm talking few studies about 2 to 3, high and low. So no one did this. Maybe it will be interesting to do systematically meta-analysis and to check the cutoffs.
Yeah, so then it will be better. You know that it will help to categorize the patient.
Meta-analysis.
And if you still have the samples, because you have taken serum samples, and if you have the samples and in the same patients, if you look at the iron levels with spermatogenesis, sperm concentration data, as well as embryo, I think you will have a lot of information. Yes.
It will be a new paper.
But the thing is, there will be a difference between ORP levels in serum and seminal plasma. So if you need to get more information, you need to do redox potential in seminal plasma and not in serum.
Yeah.
Agree. Yes.
Thank you.
So Ananta, do we have any more questions?
Yes, sir. One more question is there, sir. Does high hemoglobin have any relevance with this topic as it is likely to have higher iron level? We have already discussed this.
We have already discussed
hemoglobin.
That's it, sir.
So, one more question, last question. Do you think that iron biomarkers can help in diagnose female infertility also? Is there any effect on that? Because so many females are coming with low HB levels. Mostly in India, I can say that around 10, 11 female patients are coming. 10 milligram or 11 kind of this range are coming.
Dr. Shadi, this is for you.
Yes.
We can't hear you. You're muted, Dr. Shadi. Dr. Shadi, you're muted. I'm going to tell him.
Let me call him.
Okay, are you hearing me now? Are you hearing me?
Yes.
So in our center, we haven't studied iron deficiency and female infertility, but you can find in literature many studies showing that iron deficiency before assisted reproductive techniques can affect fertilization rate, can affect the blastulation rate, can affect even implantation rate. But we don't have too much evidence. We have small sample observational studies, not RCTs showing these results. According to in our center, the policy is to correct everything before going for IVF. But we don't have too much evidence showing that we have too much correlation between assisted reproductive techniques parameters and iron deficiency.
Thank you. Ananta, do you have any
Interestingly,
question?
there are studies which actually show the relationship between iron and PCOS, elevated thing. So it is sort of falling into the same realm.
We are working in our center on vitamin D deficiency and PCOS patients and insulin
Yes.
resistance. And we are
Yes.
finding when we have vitamin D, the preliminary results, when we have vitamin D deficiency in PCOS patients, visceral
Yes.
fat is different.
Yes.
When they have vitamin D deficiency, they have more visceral fat than subcutaneous fat. We have till now preliminary results on around 200 patients. We will publish soon, but we didn't work on iron deficiency in PCOS patients.
They're talking the other way around. They're saying excess of, but then they're saying excess of ferritin. So in some dysmetabolism of iron, somewhere. So that will also be something, because all these seem to be metabolic disorders induced by something.
Yes.
And so it's something. It's nice discussion. And
so
it's a very interesting,
congratulations. To entire team, Dr. Rad, Dr. Sherry,
Yes.
Dr. Yumna.
Thank you.
And also I would
Thank you
like
for the
to
invitation.
clarify one very important factor, because what I guess some of audience is confused between the DNA dehydroxymethylation and DNA fragmentation.
Fragmentation, right.
So be clear that these are two different things, and you cannot just
completely different. Completely different things.
Yes.
Yeah. So thank you so much, Dr. George Rath and Dr. Sherry, Dr. Yomna, Dr. Rajvi, Dr. Sanjay Shuklaji. And thanks to that Bharat Serum for providing this platform to us. And maybe in next Journal Club, we will take another good topic and call the authors. And
Coming
one more
soon.
thing.
Soon.
So one more thing, I would like to invite all of you, if you can join us in our coming online conference, EMBART 6. It will be on 8th and 9th November. So I will send you the invitation for that. It will be online. It will we always do it for 2 days. It will be for 2 days online conference. So anyone can register free. There is no issue. But I want you all to join us as a speaker, as a faculty.
Thank you. Thank you.
Pleasure.
Thank you so much.
Thank you.
Very stimulating discussion. And look forward to more of your data coming in. And
Thank you.
So
Thank
George
you.
is always on that new topics. He is doing so much work on that new topics. So it is great, man. I have seen him since 3-4 years. I am following him. He is doing so many things. Yeah. So it's nice to hear you now today. Thank you.
Thank you.
Thank you
so
much.
much.
Thank you.
Thank you.
Take
Thank
care.
you.
Bye-bye.
Bye bye. Bye bye.
Right.